The nuclear envelope is also known as the karyotheca, and it consists of two unit membranes. It means the outer membrane, which is narrow, and inner membrane, and in between both these membranes there is a intermembrane space or a perinuclear space which is filled with a fluid. Now there is a huge resemblance between both these outer and inner membrane of nuclear envelope along with the um, cell membrane. It is quite similar in structure. But if you think about the outer membrane of the nucleus or outer nuclear membrane, we find that it is rough because of presence of ribosomes over it. But the inner cytoplasmic membrane or inner nuclear membrane is devoid of ribosomes, and that's why it is smooth. Now both these structures, they are formed in continuous proximation with the endoplasmic reticulum at various places. Sometimes there is presence of certain channel proteins, and in between both these structures, like two outer membrane and inner membrane, there is presence of, although it is a continuous structure, this is a circular structure which is there, but in between there is a pore. If you see the structure of a nucleus, you will always find certain pores being present within it. So these pores are present in the surface of the nucleus, and they are responsible for allowing certain substances to pass through it. Now the inner membrane is free of ribosomes and is smooth, but has a dense layer of nuclear lamina which is present, which is closely associated with the inner surface. And the nuclear lamina is a network of protein fibers which support and form the inner membrane. It contains or it connects the chromatin to the inner membrane and also keeps most of the chromosomes at the periphery of the nucleus. The presence of this nuclear envelope is quite similar to the semi-permeable membrane, or um, a semi-permeable membrane which has the presence of certain pores or channels which allow movement of various substances. Now these channels or pores can be a wide number of pores which may be present. There may various, like thousands of pores are present in the nucleus, and each pore is fitted with a complex protein structure called the pore complex, and that is the structure which is shown over here. You can see the pore complex. It is a protein complex. The pore complex projects into both the cytoplasm as well as the nucleoplasm. Now, which one is the cytoplasm? This outside area, this is the pore. If you draw a nucleus, you may make a pore like this. Now think that this area is that pore. It is in an elaborated way. So half of it is projecting towards the cytoplasmic area and half towards the nucleoplasmic area. So this one is the cytoplasm, and inside part, this below part is the nucleoplasm area. It consists of two rings, the annuli, and it has around 8 subunits which are kind of spokes. You can see the nuclear ring, and the nuclear basket is formed of 8 spokes which make a channel. This pore complex, it controls the passage of macromolecules such as like tRNA, mRNA, ribosomal proteins, sometimes enzymes and nucleotides as well. So the RNA molecules are very large and hydrophilic and cannot pass through the lipid bilayer, and that's why there is presence of this nuclear pore which allows the movement of certain substances, those substances. So therefore these pores are quite necessary for the passage of substances which cannot diffuse and pass through the plasma membrane, or sorry, the nuclear membrane. So the nuclear envelope disappears for some time during cell division, and that is the time when we do not find the presence of nuclear envelope. Other than that, we have the presence of nuclear envelope throughout the time period nucleus is working or is present inside a cell. Now the origin of nuclear envelope, it is it does appear just after telophase or during the telophase, and it is formed with the help of small vesicles which come and join together and fuse. These same vesicles disperse or break off when it is the prophase, because the nuclear envelope, it has to disintegrate to allow the chromatin material to be suspended within the surface of the cell. There are various functions of this nuclear envelope. The nuclear envelope maintains the shape of the nucleus. It keeps the nuclear content at a particular place, at a fixed place, and well separated from the cytoplasm. It also regulates the flow of various materials into the nucleus, which are not capable of passing through the lipid bilayer of the nuclear, both outer and inner membrane. It has those pores, the central pore, which allows tRNAs, mRNAs, enzymes, and certain substances which cannot diffuse through those nuclear membrane. And the nuclear envelope itself is the site for protection of genetic material inside it. So these are some of the functions of nuclear envelope.